go. Okay. All right. So this is where we left off yesterday. It's kind of the third day we've done protein synthesis, and we've gotten to this chart. All right. So I want to start by, I'm going to pull this up a little bit, and we're going to draw a little bit up here and kind of, let's let it go there, and make this make all the sense we can and kind of review at the same time, okay? So I'm just going to redraw this. All right, so let's start off with our DNA sequence, okay? So we start off, we'll get to this in just a second. DNA. All right, let's just say our DNA sequence is A, T, C, G, T, G, okay? What would be, if this was DNA replication, all right, not protein synthesis, but DNA replication, so we need to make a complementary side, what would be the other side of the new? Oh, hang on. Okay. What would be, so you guys tell me, what is A pairs with T? Very good. T pairs with G, C, A, C. Awesome. Okay. So that's DNA replication. All right. So we've replicated the DNA. Now, what we've been talking about is not this. We're not replicating the DNA. We're making proteins. Okay. So we want to do transcription, then translation, which is ultimately making proteins. Guys, let's put those away, please, so we can not do that. Okay. So all right, the next step is messenger RNA. All right. So what would be my messenger RNA? How would this, what would I get from what would my messenger RNA be? T pairs with, okay, A pairs with U. Yeah, U, right? Because there's no T's in messenger RNA. All right, let's keep going. C to G. Or G to C, yes. C to? Okay, good. All right, so we've gotten, so this would be called transcription. Okay, that's transcription. We made the messenger RNA transcript. All right, next step is translation. All right, where we code for the amino acid. So before we code for the amino acid, I think yesterday in our notes we got to, did we get to the word codon? Yeah. Okay. So a codon was a 3-base sequence, right? AUC would be. Okay. We'll do it here in just a second if we can get to it. And the next codon would be GUG. Okay? All right. So those are our two codons. So these are codons. C-O-D-O-N-S. All right. So now this is where this chart comes in. So let's use this chart. All right. So this first one is AUC. I'm going to pull this back down so we can see it. AUC. Okay. So if I use this chart, the way I use it is I work from the inside out. So the first letter of my 3-base messenger RNA sequence, the codon, is A. So I'm going to start here. So this tells me I've got to be in here. Does that make sense? I got to be in this bracket. It's one of these amino acids. Okay? That's what this first codon codes for. AU, so I go to here. If it would have been AG, I would have gone there. So it's AU, and then C, so it's isoleucine. Okay, so isoleucine, so I could put this. I could just put ISO-L-E-U. Okay, isoleucine. It's going to abbreviate it. All right, what about GUG? You tell me, which one's GUG? G. Maybe, what's it start with? Letters it start with, instead of saying V. Yes, Valine. Good, you might want to say that whole word. Some of these are difficult to pronounce, so I haven't had exposure to these words. So G, U, G, so you got to be here. Okay? So our second one, and we'll do some more, would be Valine, V-A-L, abbreviate. Okay? All right? Let me just give you one here. So you tell me what the amino acid would be. These are the amino acids. There's 20 of them. What about UGA? Georgia. No, UGA. Stop. It's a stop codon. That's not an amino acid. That's right. Stop codon means that translation is over. Right? So when that stop codon comes around, what it means is there's no amino acid, there's no transfer RNA that pairs with that codon. So it's over. The amino acid, the polypeptide can be released. Translation is done. Okay? So that's stop. So that's how you do that. It's actually pretty easy. You just got to know how to use it. It looks like, whoa, that's like, what is that? But then when you learn how to use it, you're like, it's actually pretty easy. It's not that bad. Okay? So demystify this thing. All right. Cool. All right, guys. So what we said here was this was our amino acids. So in our growing polypeptide chain, we had isoleucine and valine were the 2 amino acids. So what we want to be able to do is do something like this on our test. Okay? We want to be able to do something like this on our test. Is you to be able to tell me the DNA, tell me the messenger RNA, tell me the codons, tell me what amino acid will be put in. You have this on the test. This same exact chart will be on your test tomorrow. Okay? All right, let's continue with these last couple pages or slides. Where did I put that clicker? I see it, the little black clicker. No, it's okay. I'll just push the button. No big deal. Here it is. All right, next one, guys. Okay, so I'm going to put this up here for a good example for you. This is in your notes. So go ahead and you can fill this in. This will be good for you to do on your own. But it also gives you a place to reference back. Okay? So, guys, this is just what we did on the board. Here's my DNA, my messenger RNA. Then we took the messenger RNA and broke them into 3 base sequences, which we called the codon. Now, I want to talk about the anticodon a little bit because this confused last period. So I want to talk about that to not let it confuse you. And then here's the amino acid. The amino acids are coded for by the codons, not the anticodon. So I'm going to be quiet while you finish writing. Good? Almost good? All right, so this is what confused last period, is this anticodon spot. Okay? So I'm going to do some artwork. I'm going to raise this up. And can you see that good enough to finish it? Okay. So I'm going to erase our thing here. Okay. Do some really quick artwork for you guys and just kind of make this. Okay. Okay, so guys, this was this oddly looking thing right here. Does that remind you of anything from yesterday? It does kind of look. It's a 737 right there. Good job, Gavin. No. Okay, that is a transfer RNA. These are tRNAs. Okay. All right. What do the tRNAs bring to the ribosome? The tRNAs bring to the ribosome, you remember? Amino acids, all right? They're right here. They're right there, okay? I'm going to draw a couple right here, okay? So those are the amino acids. So these are amino acids, all right? This big blob right here, these two big blobs are my... Not a ribosome. A ribosome, yes, good. All right, so on the ribosome, what is this? 1, 2, 3. 1, 2, 3. 1, 2, 3. What are these 1, 2, 3 base sequences right here? They're called codons. All right, so let's just make up a codon. Let's just use these. AUG. Yeah, they're called codons. So that's why it's important for you to speak and put these words out there, because when you're speaking, you're thinking, right? And it's important to try and be willing to be wrong, right? That's a huge deal, as we always say. CGA, UCA, okay? All right, so we've got these tRNAs that are coming in. So what I'm going to ask you here is, if the codon is AUG, well, what do you think the anticodon is going to be? What's this going to be right here? It already tells us up on the board. But that's what confused them, is they were trying to use this to code for the amino acid, but it's the codon that codes for the amino acid. So what this is for is so the transfer RNA, the correct one is brought in, right? So if this is AUG, then this is UAC, and that is literally the pairing of the tRNA, the anticodon with the codon on the messenger RNA is how we get the correct amino acid brought in, right? Because when you get a protein made, remember this is a polypeptide being made, which turns into a protein. Remember what we said? A polypeptide is what we call it when it's just a whole bunch of amino acids linked together. But once this thing is done and you get this like big long chain of amino acids, those are circles, bad circles, this is going to fold up. Remember what we said about the wall? If the wall is like amino acids, if you just take all the cinder blocks in this wall and broke the wall down and just put them end to end in a big long chain, it's the exact same stuff as the wall is made of, right? But is it a wall? No. Same thing as a protein would be, right? A protein is a big long chain of amino acids. But until it's folded correctly, it's not a protein. Just like this, if you just have these cinder blocks and you put them in order, until you stack them up correctly, it's not a wall. So it's kind of the same way as a protein. I'm just trying to make that make sense. It's like the protein functions when it's in the right shape. Same thing as a wall. Functions when it's in the right shape, or it's not a wall. You got to build it correctly. Okay, and not only does it have to be in the right order, it's got to be stacked in the right shape. Okay, all right. So that maybe makes a little sense for a polypeptide becoming a protein. That's what it's all about, is proteins. Okay, so this picture, the reason we drew this picture is to try to make sure we understand the difference between the codon on this is messenger RNA right here. Okay, to make sure we understand the difference between the codon and the anticodon on the transfer RNA. Does that make sense? All right. Okay. So the anticodon pairs with the codon to ensure we have the correct amino acid brought in. Okay? Because different amino acids are going to make that protein fold differently. If I put in a leucine that was supposed to be a valine, that's going to change the shape of that protein. Thus, it's going to change its function. It's not going to function properly. If the protein doesn't function properly, that's a big deal. Okay? Something's not going to work. Like sickle cell anemia. The hemoglobin is misshapen. The red blood cell is misshapen. The red blood cell doesn't carry oxygen as good as it should, or it would normally if it wasn't misshapen. So that's a big deal, right? Somebody who has sickle cell anemia, they can't do sports, or it's hard to breathe at high altitude. or they can do sport, but they got to have it. It's harder for them, you know, because it's hard to transfer oxygen around your body. Okay, let's continue. All right, next one, and guys, we're almost done. Hey, this slide right here is a great summary, and then we're done. Okay, so let's put in this information right here, guys. Let you fill in that slide, and like I said, this is a great place for you to go back and study, and then on the next slide, I just want to ask one question for the class to contemplate, for us to think about a little bit and see if we can come to an answer for a question that hopefully maybe gives you an aha moment, that sort of thing. But just fill this in because it's a great place for you to study for your test tomorrow. So as you're writing, guys, just to talk through this briefly, if we go down from the top down, it gives you the purpose, the building blocks, the location. So, you know, easy test question here, you know, simple test question, but kind of, you know, it's a simple test question I asked, but it's just a recall question, which can be tough if you don't remember it. All right, so the location of DNA replication is the nucleus. But hopefully, like we said, if we're painting a picture in our mind and making it more like a story and not just remembering random facts, it'll hopefully help this become more than just recall. You can picture a cell. You know where the DNA is located. If it's DNA replication and you're picturing the cell in your mind, you could picture that the DNA is located in the nucleus. So that has to be where DNA replication happens. You know that transcription takes place in the nucleus because that's where the DNA is. And transcription is DNA turned into messenger RNA. So if you can picture that process, you can remember it a little better. And translation, you know that that's where proteins are made. Okay. Well, are there any ribosomes in the nucleus? No. So you know that that can't be where translation happens. So you know it has to happen outside the nucleus on a ribosome. Either in the cytoplasm or the rough ER. Right? That is the last slide where we're going to write anything today. But on the next slide is a question for the class. I want to try to answer it, okay? So question for the class. Guys, contemplate this question. Think it through. Be willing to answer and get it wrong after thinking it through, okay? Read that question. Take all the time you need. I'm going to start my stopwatch in about a minute to 90 seconds. I want you to try to answer it. I'm going to give everybody about another 15 seconds, and then Grayson will be the first one to give us a shot here. And others can add on more than likely to Grayson's answer if you've got something else. Okay, Grayson, what you think?
Because some of them has DNA in them, and then the others have mRNA or RNA.
Okay. Okay, think about it. What Grayson said was some of them have DNA in them and some of them have messenger RNA in them. And I'm so glad he said that because that's going to lead into some really good discussion. So just go that way, guys. Okay. So, guys, do any of our cells not have DNA in them? No, they all have DNA. Now, if you want to get down to the particulars of that, there's actually some cells when they become mature, they actually lose their nucleus. But we're not going to discuss that. In general, yes, they do. They all have DNA. So does that mean that all of our cells have messenger RNA? Are going to make messenger RNA? Yes. Okay. So, but you're thinking, talking's thinking. Okay. So somebody else kind of add on to that. I think, Hayden, did you have something to add on to this?
I was going to say that they have different codons.
All right. Well, okay, you're leading into the right answer. They have different codons. All right. Where do the codons come from? What are the codons located on? Okay, the ribosome is a big protein and ribosomal RNA complex that is responsible. It's the machinery responsible for putting together the amino acids. It gets everything together, makes the bonds happen, and builds the polypeptide, right? Okay, so Hayden, let's stay with me. So the codons are located on the messenger RNA. Okay, all right. Okay, it's all right. So the question, though, is let me state the question for us again. If all of our body cells have all of our DNA in them, how come my cells are so different? For example, why does if my skin cells have the same DNA as my muscle cells, why are they so different? Why do skin cells look like this and why do muscle cells look like that?
Because skin cells have proteins and muscle cells don't.
They both have proteins. Remember, they all have proteins, but you're hinting at the right thing. So first off, okay, all our cells have all our DNA. But do you remember what we said? How much did we say of our DNA is made into proteins? Do you remember the number, roughly? What percent of our DNA would be called genes? Remember that? I just mentioned it in class. No. Okay, let's say it right now. Only about 3%, guys, 2 to 3% of our DNA are made into proteins. That should be extraordinary. That's extraordinary to me. So you're telling me that 97% of the instructions aren't made into flesh and blood? That's what I'm telling you. That's nuts. First off, that's just nuts. So only 3% are made into flesh and blood. So out of that 3% that get made into flesh and blood, now we're learning more about what the other 97% do, right? They're regulatory sequences, all kind of stuff, but they're not protein. They're not genes. All right. So, but we still haven't answered our question. Zach?
Does each cell use different parts of DNA?
Yes. Yes. Now, Zach's back there. He's just contemplating, man. Like, Zach, that's right. That is right. It's because each cell uses different parts of the genome. That's correct. It's like a cookbook, guys. When you make dinner, do you make every recipe in the cookbook? No. Good gracious. You can't do that. You can't do that. You make one. There might be hundreds of recipes in that cookbook. You make one recipe, right? So as a heart cell, you only make maybe the recipes that are different types of spaghetti. As a lung cell, you make the recipes that are different types of desserts, okay? So you only use a small section of the code, and you make the proteins that are specific to you, just like Zach said, okay? Because remember, what do cells do? When they begin, we said this is putting all the puzzle pieces together, man. This is where this beautiful picture of protein synthesis and starts to build. Remember, we're about to stop. When we started off, right, we started off with fertilization. Sperm fertilizes egg. You have a cell that can become anything. It's undifferentiated, right? Remember that word differentiation? So that's what starts to happen. It's literally what starts to happen is cells differentiate. The code is the same, but literally certain parts of the code start to become unusable by the cells as they differentiate. They can't use that code anymore. So it's like when we said our cookbook analogy, it's like you can't read these recipes anymore. It's like a little lock has been put on this part of the recipe. And you can only use the recipes for making pizzas. You know, that's all you get because you're a pizza making cell now. Okay? But the brain cells, they can make, this fancy recipe. They can make I don't know, pasta. Yeah, pasta. Okay, pasta. They can make that from scratch. It's kind of hard to do. Yeah, But that's all they can do, though. Now, there are some cells in our body, we call them stem cells, right, that still have they have a lot of the cookbook left, but not all of it. All right, they have a lot of the cookbook left, but not all of it. And so that's really putting all that stuff together, okay? So, all right, so let's, are we done? We're going to play Kahoot. So if you got computers, get them out. Okay? Yes?